Topic of discussion is bacteriophage. What is a bacteriophage? The bacteriophage is a type of virus that infect bacteria. It is a type of virus that infect bacteria. These are called phages. The word bacteriophage literal meaning bacteria eater because bacteriophages destroy their host cell. So literal meaning bacterial eater, it destroys their host cell. They can replicate only in bacterial cell. They are obligate intracellular parasite that multiply inside bacteria by making use of some or all of the host biosynthesis machinery. They are ubiquitous in nature, presented everywhere, and are recognized as the most abundant biological agent on earth. They are extremely diverse in size, morphology, and genomic organization. They are diverse in size, morphology, and genomic organization. With the help of electron microscope, scientists studied the detailed visualization of hundreds of phage types, some of which appear to have heads, tail, and leg. These phages have tail and leg, but they are non-motile. By using Brownian movement only, it will reach to the target places. What is Brownian movement? Here a lot of phages is there. So lot of phages to push one by one to reach that target place. Name is called as Brownian movement. These pouches used in basic molecular research and epidemiological tracking. So they have been used in molecular research and epidemiological tracing. Epidemiology means study about disease. So this is all about bacteriophages. Ernest Hornberry Henkin in the year of 1896 reported on the Ganges and Yamuna river in India. They first reported these bacteriophages in Ganges and Yamuna river in India. They stated water seemed to have some sort of antibacterial property that helped against bacteria, that cholera. This Ganges and Yamuna river water have antibacterial properties against cholera. Then 1917, D. Herle and his co-worker isolated with lytic activity against pathogenic bacteria. What are the bacteria? Escherichia coli, Neisseria meningitis, Pasteurella, Pseudomonas, Salmonella typhi, Shigella, then Streptococcus, Vibrio cholerae, and Yersinia. He developed an idea of phage therapy as prophylactic, that means preventable, or therapeutic use of selected bacteriophages in the destruction of pathogenic bacterial cell. So in 1917, T. Here and his co-worker isolated bacteria. Then there are using this one for phage therapy. or destruction of pathogenic bacterial cells. Next, types. Bacteriophages are two main types based on the replication, that is virulent phage. Second one is temperate phage. So first one is virulent bacteriophage types. These virulent bacteriophage types used as lytic cycle for replication. These one use the lytic cycle for replication. Lysis or lytic cycle is a cytoplasmic viral replication process in which the bacteriophage inject its genomic material into the host cell. In this viral replication, this bacteriophage inject its genetic material to the host cell, so to that bacteria. This allowed the replication process to begin producing many phages. After starting replication, it producing many phages. Once the host cell is filled with a new bacteriophage, the host cell rupture from within and releasing the newly formed phages. So once ruptured, it will releasing newly formed phages. It is important to note that bacteriophages that are used for phage product and phage therapy. So these newly formed phages used for phage product and phage therapy. Now we are using that phage therapy. All are virulent phages. All are virulent phages. So virulent bacteriophage uses lytic cycle for the replication. Bacteriophages inject its genomic material to the host cell. After that replication process started, it producing many new phages. Once it filled into their host, after that the cell ruptured and releasing newly formed phages. This one used for phage therapy. All the new phages are virulent. Second one is temperate bacteriophages. Temperate bacteriophages use the lysogenic cycle for its replication. This cycle allows the phage to place its genome into the bacterium. So second one is temperate bacteriophages. This one using lysogenic cycle for its replication. So lysogenic cycle for its replication. This cycle allows a phage to place its genome into the bacterium, but instead of rapid replication in lytic cycle, they started rapid replication. But here this genome referred to a prophage. Prophage means dormant state. This prophage either integrated itself to be a part of host chromosome or may remain as a plasmid in the cell. So here this lysogenic bacteriophage remain in this symbiotic state. Symbiotic state means not harm to the host cell, but rather quietly uses the resources provided by the host cell. Here not harm to host cell, but it can uses the resources provided by the host cells. However, when a lysogenic bacteriophage feel under a sudden amount of pressure or it survival is placed as risk, it has been ability to switch from the lysogenic cycle to lytic cycle, which result in rapid replication of newly formed phages which burst out of the host cell. So here their lytic cycle rapid replication started, but here it become prophage. Prophage means it integrated to be a part of host chromosome or remain as a plasmid cells. So here this bacteriophage symbiotic state, that means not harm to host cell, but it can use resources from that host cell. This lysogenic bacteriophage feel that survival is difficult, means it have ability to change lysogenic to lytic cycle. So here rapid replication process started, then newly formed phages burst out of that host like lytic cycle. Now, characteristics of bacteriophages. Bacteriophages have high range of host specificity. They usually inhabit the intestinal microflora of human and animals. They presented in intestinal microflora of human and animals. Bacteriophages have a single standard DNA or double standard DNA, single standard RNA or double standard RNA genome. Okay, these are the genome has been identified. Bacteriophages genome consists of four genes, genus, or it may be several hundred genus. So it consists of few as four genes or many as 100 genes. Like most viruses, bacteriophages typically carry only genetic information needed for replication for their nucleic acid and synthesis of their protein coat. Lytic phages lysis bacteria during active phase of bacterial growth. The capsid of bacteriophage can be isosahedral, filamentous, or head-tail in the shape. The head-tail shape seem to be unique to the phages. Phages are inactivated by boiling. So these are the important characteristics of bacteriophage. They presented in intestinal microflora of human and animals. They may be single standard or double standard DNA or RNA as a genome. That fews are 4 gene, many are 100 genes. Then they carry genetic information needed for replication of that nucleic acid and synthesis of protein coat. Lytic phages lysis bacteria during active phase of bacterial growth. Then it have isosahedral filamentous head tail in shape. Maximum unique structure means it have head and tail. Then inactivated by boiling. Next, structure of bacteriophages. Most of the phages consists of single linear double standard DNA genome. So most of the phages single linear double standard DNA genome. The phages have simple structure comprising genome which is surrounded by a protein covering is known as phage capsid. Large phages usually consists of head and tail. So large phages consists of head and tail. The head encloses the genome. Head encloses the genome and the tail is used as an organ of attachment as well as conduit through which phage DNA passes into the host cell. By using this tail only, it will pass that DNA genome to the host cell. The best studied phages are the T1 phages, T2, T4, T6, etc. that infect the bacteria Escherichia coli. So by using this tail only. It carry the passes the DNA to the host cell. These phages are traditionally considered as a prototype for describing the morphology of bacteriophages. Phages are tadpole safe with a hexagonal head and cylindrical tail. So most of the phages single linear double stranded genome. It have head and tail part. Head enclosed genome, tail used attachment and to pass the DNA to the host cell. Best studied T-even phages T2, T4, T6, etc. They infect Escherichia coli. Phages are tadpole safe, with hexagonal head and cylindrical tail. Now discuss about the structure of bacteriophage head. All phages containing a head structure which can vary in size and shape. It is isosahedral, 20 sides it have, filamentous or head tail type of shape. The size of the head is measured between 28 to 100 nanometer in length. The capsid of bacteriophage head encloses a compartment that containing nucleic acid, which is most of the phages is double-stranded DNA. The capsid or protein coat of a virus is composed of individual protein is known as capsomer. Next, tail. Many but not all phages have tail attached to the phage head. Not all phages having tail. The tail consists of hollow tube through which the nucleic acid passes during infection. Coming to importance of bacteriophages, they are convenient model for study of virus-host interaction. They play an important role in transmission of genetic information from one bacterium to the another by the process of transduction. An important example of this is drug resistant seen in Staphylococcus aureus. So, what is the important role? Transmission of genetic information from one bacterium to another. Here, this Staphylococcus aureus is the drug resistant. So, it is an important because of this bacteriophage only. This Staphylococcus aureus is the drug resistant. Then they are used as a cloning vector for genetic manipulation. Then they are used for phage typing. Phage typing is used as an important epidemiological marker which help in establishing the path of transmission of infectious agent and identifying the reservoir of infection. Then bacteriophages may provide the property of toxigenicity to some organism such as Corynebacterium diphtheriae. Bacteriophages are also important in industry and medicine. For example, many phage destroy the gram-positive lactic acid bacteria that are critical to the production of fermented milk products such as yogurt and cheese. Bacteriophages can carry variety of virulence factor that convert that bacterial host into potential pathogen. This this is the cases for major human pathogen such as Streptococcus pyogen, Staphylococcus aureus, Corynebacterium diphtheriae, Vibrio cholerae, E. coli 0157H7. On the positive side, Russian physicians have used bacteriophages for years to treat bacterial disease. For many years, they are using bacteriophages as a therapy. Research studies indicate the phages may be effective in treating bacterial infection, including those caused by antibiotic resistant bacteria. So, this is all about bacteriophages. Thanks for watching.